Hello. So the topic today is immunology, and we have a question here for grade 2000 and from grade 2019 biochemistry for 2 months. So the question asked here is that a mixture of the following purified proteins IgG and FAB fragment of the immunoglobulins is separated using gel filtration chromatography. The order of elution of these proteins is. So there are various options, and to answer this question, we need to know what are firstly what are immunoglobulins, and we also need to know what is gel filtration chromatography. So first, we'll understand the basic concept behind the different immunoglobulins, how and also the FAB fragment, and then we'll study about the gel filtration chromatography. So immunoglobulins are basically B cell antigen receptors which are produced by the B cell or the B lymphocytes, which are a class of the white blood cells. B cell antigen receptors are present on the membrane of the B cell. They are attached on the membrane. They have a transmembrane region which helps them to remain attached onto the B cell membrane. Now, this B cell antigen receptor, has a peculiar structure. So, it is composed of two heavy chains. These long ones are the heavy chains which have the transmembrane region, and they also have two light chains which are associated with each light chain is associated with the heavy chain, and they are joined together by the disulfide bridges. So there are three or four they are disulfide bridges which join the heavy chain to the light chain, and one disulfide bridge to join the heavy chain with the heavy chain. So each chain has a variable region as well as a constant region, both the heavy chain and the light chain. The variable region from both the heavy chain and the light chain, they together form the antigen binding site. So there are two antigen binding sites in one immunoglobulin. It will have two antigen binding sites. So another important thing to note about the antigen receptors is that they are very specific. So a B cell will only produce one kind of antigen receptors. So as you can see, this B cell has antigen receptors whose structure are same. So it will recognize only a particular kind of epitope. So what are epitope? Epitope are a small region of the antigen which is recognized by the B cell receptor. So if this whole thing is an antigen, epitope is the small region. So here you can see an antigen consists of various epitopes. One can be circular kind of or conical kind of or others, and this B cell will recognize only one particular kind of epitope. Then it will get activated. After recognizing the epitope, it will get activated, and then it will start secreting these antigen receptors, which are then called the antibodies. These antibodies will recognize those particular epitopes, and that will help in further destroying the foreign particle or the foreign organism. And there are various antibodies like say suppose antibody A, B and C, and antibody A will recognize only one particular kind of epitope. Say suppose this conical one. Antibody B on the other hand will recognize this circular epitope, and antibody C will recognize a different kind of epitope. So each antibody is very specific, and it will recognize only one particular kind of epitope. Another important concept to know here is that this heavy chain and light chain can be digested in various ways. So, this heavy chain and light chain, as you know, they are composed of the disulfide bonds. So, if we perform beta mercaptan ethanol reduction on them, so these disulfide bonds will get reduced, and we will get individual heavy chains and light chains. But if we perform pepsin digestion, so pepsin is an enzyme which cleaves after the disulfide bonds. So we'll get this kind of structure which is called the FAB prime 2, in which the variable region of the heavy chain and the light chain together, they are joined together like this structure, and we will get two Fc fragments, individual Fc fragments. And but if you perform a papain digestion, it will cleave above the disulfide bond. So what we will receive is that after digestion is two FAB fragment like this. They are joined together by the disulfide bond, and two FC region which are also joined together by the disulfide bond. So this is what we will get, and here you can see as a three-dimensional structure of the immunoglobulin IgG. Now we will come to the different classes of immunoglobulin. So there are five different immunoglobulin classes, and they are differentiated based on the composition of the heavy chain as well as the number of Ig domains, that is the constant Ig domains. So first of all we have IgG, which is composed of the heavy chain gamma, and it consists of three constant regions. IgM, composed of heavy chain mu, it consists of four constant domains. IgA, composed of heavy chain alpha, consists of three constant domains. IgE, composed of heavy chain epsilon, consists of four constant domains. IgD consists of heavy chain delta, and it has three constant domains. So, and each of these immunoglobulin classes or the heavy chain can associate with the light chain kappa or lamin. So light chain is common, and but it is the heavy chain which is the differentiating factor. And also there are some of the immunoglobulin consists of a J chain or a joining chain. These are present in IgN as well as IgA antibodies only. And why are they present? Because IgG, D, and E, which do not consist of a J chain, here you can see IgG, E, and E do not consist of a joining chain, they exist as monomeric form only. IgG, D, and E. But IgA and IgA, as they consist of a joining chain, they can form higher molecular weight structures, and IgA can exist as a dimer in this form where the two monomers are joined together by a joining chain. IgM can exist as a pentamer where the five immunoglobulins are joined together by the disulfide bond as well as the joining chain over here. So after having a brief knowledge about the antibodies, we read about the gel filtration chromatography. Chromatography is a technique where we can separate different substances. And gel filtration chromatography in particular is a technique which can be used to separate out proteins based on their molecular weight. So there can be different proteins like of different globular proteins. So and they would have different molecular weights. Gel filtration chromatography helps us to separate out different proteins based on the molecular weight. So the basic principle behind gel filtration chromatography is that we have a column in which we add our gel or the beads suspended in a buffer, and then we load the sample mixture which consists of different proteins or different molecular weights. Now this sample, it passes through the column and it gets separated. How? The gel beads has small pores. Okay. And these pores are of a particular size. Now, the bigger proteins cannot enter these pores. So, the molecule with larger molecular size eludes with the mobile force. So, these bigger proteins will not enter the pores, but the smaller proteins, they get trapped inside these pores, and their movement is retarded. So here you can see these are the kind of pores in the gel beads, and there are different proteins whose size is according to this whose size varies. So the bigger proteins, as they are unable to enter inside these pores, they will bypass these pores, and they will move through this kind of route, okay, in the mobile phase, and they will elude the first. Here you can see the bigger blue proteins, they do not enter the pores, and they are the first ones to get eluded. Then we have the medium-sized proteins which are somewhat retarded by the pores, and then they will elute after some time after the bigger protein has eluted. And the last protein to be eluted are the smallest proteins because their movement is retarded the maximum by because they get trapped inside the pores of the gel beads, and so they come out the last. So in gel filtration chromatography, the protein which has the highest molecular weight will elute the fastest, then with the medium, and the protein with the lowest molecular weight will elute the last. So, based on this information, we can answer this question. So, we have a mixture of purified proteins which are IgG, IgM, and FAB fragment. So, as I have already mentioned, and I will again mention that Ig, G, E, and D, these all immunoglobulins, they exist as a monomer. Okay, they exist as a monomer, and Ig it exists as a dimer, and IgM it exists as a pentamer. On the other hand, FAB fragment is there, which is the cleaved, which we get after cleavage of the immunoglobulin by the enzyme PAPin, we can get FAB fragment. And so, according to this figure, the highest molecular weight will be of that of IgM because it exists as a pentamer. Then we will the next protein will be IgG, E or D. No, sorry. The next protein will be IgA, which is because it exists as a dimer. Then we'll get Ig, G, E, and D because they exist as monomer. And lastly, we'll get the FAB fragment because they are the smallest. So, based on this information, the protein which gets eluted from first to last. So, the protein which gets eluted first is the one which will have the highest molecular weight. So, we normally depict the elution in the form of a graph. So here it is the time of elution. In the x-axis we denote the time of elution, and here we denote the intensity or the amount. So the protein which gets eluted first will be IgM because it has the highest molecular weight, and then we will have IgG because it exists as a monomeric form, and then lastly we will have not Ig, FAB fragment because it is the smallest uh, of all these proteins. So IgM first, IgG second, and FAP fragment third. So the correct option is option number B. So here two things you have to know because it is a gel filtration chromatography. Order of elution will be the of the highest molecular weight to the lowest molecular weight, and here it is written from first to last. So they can play with this term. So they can also write from last to first also. So these two things you have to keep in mind and then answer the question. And the correct option here is option number B.